So last week we talked about prokaryotic gene expression. Today, eukaryotic gene expression. This can get really complicated really quickly. We're going to try and keep it short and simple. AP doesn't hit it too hard. There's just a couple of things that they tend to hit from time to time. So we'll make sure we cover those. So a lot of this presentation is going to be details that I normally teach, but we don't need to get into all of them. So I'll probably go through it kind of fast. If I go through it quickly, it's something you don't need to worry about. I'll tell you at the end and throughout what are the really important things. And these first few questions, this first couple of slides, these are the kinds of questions they like to ask a lot. They compare prokaryotic and eukaryotic gene expression and how we accomplish the same things. So first of all, there's no operons in eukaryotes. And the reason for that is that they are too simple. To turn on all those genes at once, right? We use genes in a lot of different ways. Like the third gene needed to synthesize tryptophan might also be used somewhere else, in some other cell to do some other thing. And so we would have to turn on all of the tryptophan genes to activate that gene again. So, that's not going to be effective for us. We're a lot more complex and a lot more, we need a lot more control over each gene's expression. But we do have the ability to turn on a bunch of genes at once. We just don't use an operon like that. To accomplish this same thing, we use transcription factors. A transcription factor may be able to bind to a number of different sequences of DNA. And so in different cells, it can be used to help turn on a lot of different proteins. And in some cells, there may be other transcription factors that block, a certain transcription factor. So again, it's that combination of transcription factors that finally summons the RNA polymerase and allows us to transcribe this gene. So each individual gene has its combination of transcription factors that will bind it so that it can be transcribed. Not only that, but which transcription factors bind it also determines how much we'll transcribe. You'll see that in a minute. And of course, this is what makes cells different, right? The difference between a liver cell and a brain cell, the reason they're a different shape, the reason they have different proteins, is all about gene expression. It's which genes are turned on. And so when you turn on a certain set of genes, that cell becomes a pancreatic cell. You turn on a different set of genes, that cell becomes a muscle cell, right? It's all about which genes are being expressed. That's what makes one cell different from another. Depending on how you want to count them, we have 7 different points of control, 7 different ways we can control how much of a particular protein is present in a cell. Some of these AP never mentions, some of them AP hits from time to time. So we need to make sure you understand a few of them really well, and the others we can kind of let them go this time. So first of all, you got to keep in mind that your DNA, when you pack it up into a chromosome, right, it almost never looks like this. It looks like that for mitosis, and that's all. But there are parts of it that remain packaged like that, and depending on what cell it is, it's going to be different parts, because the parts that are bundled up like that, there's no access to those genes. Transcription factors, RNA polymerase, they cannot bind. So the DNA wraps itself around these proteins called histones, and histones come up from time to time in AP. Histone proteins have R groups that are attractive to DNA. They bind to the DNA. And if you think about the structure of the DNA with that double helix and what those molecules are on that outside edge, you'll remember it's a sugar and a phosphate. Now, phosphate has a negative charge, so DNA has a negative charge. So we're talking about positively charged R groups. Histone proteins have positively charged R groups on the outside that attract that DNA and allow it to bundle up around these things. And so one of the ways we can regulate whether or not a gene is going to be expressed is how tightly we have it bundled up. So it can be bundled up tightly, or we can acetylate the protein. And the thing we're actually going to acetylate here is a positively charged amino acid. Lysine is the amino acid. It's positively charged. We stick this acetyl group right onto the spot that has the positive charge. So the positive charge is now gone. So it no longer has that positive charge. So when we acetylate histones, which we have enzymes to do that, that makes it so that the DNA kind of loses this tight bundled structure and kind of opens up a little bit so that transcription factors and RNA polymerase can get to it, and we can use the genes that are in that stretch of the DNA. So histone acetylation is a way to upregulate, which basically means express more of this protein. So we use this as one of our primary ways of controlling gene expression, bundling or unbundling parts of the genome. There are whole sections of the genome that contain things that you don't need in in a cell in your retina, and those will just stay tightly bundled up for the entire life of the cell. Other parts that you have to have a lot will have acetylated histone so that it can be unbundled, and we can have access to the DNA strand to that sequence so that transcription factors and RNA polymerase can do their job. A couple of vocabulary terms associated with this. It's not something you really need to worry about. But euchromatin is the part that we are expressing. Heterochromatin is bundled tightly. It stains darkly because of this tight bundling. DNA methylation changes the shape of the actual DNA double helix. You're going to stick a methyl group on a cytosine, right? So you're seeing here's cytosine, and we're just sticking a methyl group on there. A CH3 is a methyl group. And that changes the shape of the molecule, and therefore it changes the way proteins interact with it because it's all about shape, structure, and function. Things like transcription factors, things like histones, they interact with this differently. Now, this is like a change that can be made to the actual DNA sequence that makes it so that we can downregulate or reduce the expression of a particular gene. And what's really interesting about this is that these are heritable. Methylation, DNA methylation is heritable. So if your parents had methylated DNA, one of your parents had methylated DNA, you get that too, which means that gene is going to be expressed a lot less in you than it would be in people who didn't have that gene methylated. And it's interesting because this is almost like the one tiny little example when Lamarck was right, because you can acquire methylations during your lifetime. Certain events that occur, like starvation, extreme starvation can cause some of your genes to be methylated. And it's genes that make you less efficient in burning calories. So it's a starvation response to shut these off so that you don't waste energy. And you can inherit that later. So it ends up like certain populations we've discovered that have gone through a severe starvation period, their children and their grandchildren have problems with obesity because their genome has been altered so that they conserve energy and save it as fat. So it's this one little example where it seems like an acquired trait was inherited, which is what Lamarck was talking about. So was he still mostly wrong about that? Yeah, but there's a little example where he wasn't. Methylation of DNA is part of what we call epigenetics, which is molecules that bind to the DNA and change the way things are being expressed. And there are a variety of epigenetic factors, things that can bind to this, but a methyl group is one example. So we also don't have anything in a eukaryote that's as simple as an operator where you can just stick one protein on and this gene is on or off, right? That we don't have anything like that. It's got to be a lot more sophisticated. Again, what we give up in simplicity and efficiency, we gain in control. We gain really fine control on this immensely elaborate, and complex process of, keeping a cell alive. And when you think about it, right, a bacterium only has to worry about that single cell. Part of why we don't need an operator is because we can turn on a whole bunch of genes independently, and we can do it in a cell, which has a very different function than some other cell, which you can't do if you're a single-celled bacterium, right? Everything's in the same cell. And so to initiate transcription in eukaryotes, you have lots of transcription factors that are going to be involved, but there's even more than that. So we have this promoter sequence, right? We have transcription factors that bind to that, lots of transcription factors that are all going to bind to this to summon RNA polymerase. But there's even more than that. There's this enhancer region, which is farther upstream this way from where we're going to transcribe. And so the enhancer region, so here's enhancers here. And in this case, we have activators bound to them, but you can also have silencers. So you could have activators or silencers bound to these. They speed up the RNA polymerase and make it so that when one RNA polymerase goes, another one is very quickly coming and another one is very quickly coming. And we'll have multiple RNA polymerases running down this gene, cranking out RNA so we express it a lot. You can also silence the enhancers, which actually slows down but doesn't stop the expression. Removal of the transcription factors, or just not having the transcription factors there, means we can shut off the gene completely, or bundling it up like we said before. All of this can be elaborately controlled by which transcription factors, activators, silencers are all there, right? Even transcription factors can be on or off molecules. This diagram gives you a little bit of an idea of how complicated this gets. I've got these 3 different enhancer sequences with activators bound to them. I've got the promoter sequence with all of this, lots of different transcription factors bound in here, all to get this RNA polymerase here so that we can start coding, getting that mRNA or that transcript. So it's not unusual for, 10 or 20 different proteins to be involved in initiating transcription of a gene. Like I said, what we lose in simplicity and efficiency, we gain in control. We talked about mRNA splicing and exon shuffling, right? These are ways to express different proteins from a particular gene. So again, we're talking about, we can make this one, we can make this other one, right? There's different ways to do this. As we rearrange these exons, we can get different proteins. So we can make more of one or more of another. We also talked about the 5' cap and the poly A tail, right? You can shorten that poly A tail so that you only make a little bit of this protein because you're only going to use each transcript a few times. Or you can make a long poly A tail and it'll hang around for a while. We can express that protein for a long time. And of course, just removing the 5' cap allows it to be digested. There's a little process called RNA interference, which is kind of interesting. I've got a much more complicated description here than what you need, but honestly, I don't think AP, it's not even in their outline of things they'll ask about, but it is a way that we regulate what proteins are present, even after transcription. So we can go through the trouble of making a transcript and then immediately shut it off. And we can shut it off very, very quickly, which sometimes we need to do. I think we talked about before how having that 5' cap and poly A tail means that each RNA has an expiration date, but that may not be soon enough. Maybe you need to stop making that protein right now. And RNA interference allows that to happen because as soon as you express this RNA, it's going to get chopped up and it's going to bind to any messenger RNAs that are out there of that particular sequence and shut them off completely and have them be degraded. So again, this is another slide kind of going through all the details of RNAi. It is something that's being exploited as a technology now. We can shut off genes specifically. It's used a lot in research. And there are certain therapies that are being developed using this technology. This is just a diagram kind of showing how RNA interference works. You don't need to worry about the details. We can also use proteins to stop translation by binding sequences of RNA. So they can grab the messenger RNA right where the ribosome would bind and prevent that RNA from being used. So we can also regulate the expression of a gene at the RNA with proteins. And once we have made the protein, we can still edit the protein, we can change the protein, we can destroy the protein. So even at that point, the cell can still go, that protein that I made, I don't want it. I turned the gene on, I've used the transcript, I made the protein, I don't want that protein. And they can still shut that off. And there are mechanisms for doing that. And we have some proteins that we normally keep just completely inactivated, and we only activate them sometimes. You remember your whole kinase cascade. Those proteins are sitting there waiting to go all the time, and all you got to do is stick a phosphate on there, and now they're active. So we can make these proteins in an inactive form. We also talked about zymogens, same type of thing. Upregulation and downregulation, we're talking about activating the gene or producing more of the protein, that's upregulation, and producing less of the protein or shutting off the gene, that's downregulation. And if you look, right, so bundling DNA with histones, that's downregulation. DNA methylation is downregulation because it makes it easier to bundle. Transcription factors can do both up or down. Enhancer sequences are usually up, but they can also have silencers behind them, which downregulates. Poly A tail and 5' cap, those are basically ways that we can up or down regulate the amount of that stuff that's going to be there. RNAi is all down regulation. Regulatory proteins binding the transcript, right? So they're blocking translation. That's down regulation. Protein processing, right? It may be that we're changing the protein from one thing to another. So we're making more of that other one, but less of the other. And protein digestion, of course, would be down regulation too, because we're not going to use that protein anymore. You don't need to know all this. This is a lot more detailed, and I do this just for my own satisfaction. So this slide basically has all the things that I've just kind of shot through there. And just to give you a heads up, things that AP asks about. Do they ask about DNA packing and histones? They do. Histones is something that you should know about, right? When it's packed around histones, it's not expressed. That's all there is to it. Transcription factors, which we've talked about those quite a bit before. They're part of our cellular toolbox, but definitely. RNA processing, right? Splicing introns. AP is going to ask about that. 5' cap, poly A tail. That they ask about that too. They do not ask about RNA interference. They don't ask about blocking translation with proteins. And they don't really ask about any of this stuff either. So it's mostly early stuff. And that honestly is more of how we regulate gene expression than these other things. So they are hitting the high points. This is kind of an interesting pie graph. This is what I've told you many times before. 1.5% of your genome codes for exons, for segments of DNA that code for proteins. The rest of this stuff, A lot of it is about regulation. There are lots of non-coding RNAs, and that's that RNA interference we're talking about. But there's also some other RNAs that we've recently been discovering that are long non-coding RNAs. When we say non-coding, it just means it doesn't code for a protein. So like tRNA is a non-coding RNA. Ribosomal RNA is non-coding also. So a lot of those things are all included here. But a lot of that is about gene expression and how we regulate them. And we're learning a lot about that. What we're learning is that most living things on Earth, they share this 1.5%, and they're pretty similar. There are little version differences, but they're pretty similar. Where we really differ from all the other animals is in how we use these genes. We've talked about this before. Transposons are a big part of the genome. A bunch of it is introns, and a lot of it is just repeats, just the same things over and over again. And that includes things like telomeres and centromeres, which are just big repeated sequences. Hopefully that was short and sweet. We got genetic technology coming up in the next one. I'll go to school and do a couple of little labs, or at least show you some tools that we use in labs. Hope you guys are doing well. Hang in there. You're doing great.